Good morning, everyone. Thanks for joining us. I'm Dan Hurren, one of the healthcare analysts here at MST. And this morning we're joined by Dr. David Stamler, CEO of Alterity Therapeutics. Dr. Stamler has held that role since January 21 and previously served as the company's Chief Medical Officer since 2017. I think you're all pretty familiar with Dr. Stamler, so I'll keep it short. He has an extensive background in pharmaceutical development, particularly in the fields of neurological conditions and movement disorders. His career includes senior leadership roles at several prominent biopharmaceuticals, I should say, including Teva, Auspex, and just to name a few. Dr. Stamler, welcome and thanks for joining us. We do appreciate your time. Just before handing over to Dr. Stamler for his presentation, just on a point of admin, if you do have questions, please use the Q&A function you'll see at the bottom of your screens. And at the conclusion of Dr. Stamler's presentation, we'll go through those questions and interrogate the doctor. So, Dr. Stamler, thanks again. Over to you.
All right. Thank you, Dan, and welcome everyone. I'm really glad to come back and update you on the progress we've made over the last several months. It's been actually 5 months since we disclosed our top-line data, and we continue to pressure test, analyze the data, and I am as confident as ever about the strength of our results and our path forward. So I've got just about 10 to 12 slides I'm going to walk through just to orient people who may be new to the story. And I'll talk a little bit about what we've learned along the way, and then certainly happy to entertain any questions you might have. So Dan, if you would go to the next slide. So just to give you a bit of an overview, our lead indication is a disease called multiple system atrophy, and I'll tell you more about that in a moment, but there is no approved therapy for this disease. It's quite a devastating condition, so what we've shown, I think, is quite important. In January, we did disclose that we had very strong clinical efficacy data on an endpoint that really matters to the FDA. And not only did we see efficacy, but it's at the level at which others have published that shows that it is clinically meaningful. In April, we actually got fast track designation for our lead drug called ATH434. Sometimes I just refer to it as 434. And that is for the treatment of MSA. And we may, afterwards, if a question comes up, what does that mean? I'm happy to talk about that. But that does give us various benefits in interacting with the FDA. After we released the data, on the strength of those data, we were able to raise significant capital, and we are very well capitalized now to do everything that we need to do to get to phase 3 and to even prepare for the start of that study. We do have a strong patent portfolio, both of surrounding 434 as well as new compounds which could be taken forward in other diseases. And in addition to myself, I'm surrounded by a group of excellent drug developers. We've been working together for over 15 years. And I think we're a very high-functioning team, and we've been quite successful. So I'm really happy that we're continuing to work together on this important disease. So next slide, Dan, we'll wanted to set the stage and talk about our overall strategy. So we are targeting neurodegenerative diseases that have iron related to their pathology. And this is not all neurodegenerative diseases, but it's a lot of very important ones. And you see some of those pictured on the right. Parkinson's disease is kind of at the core. And what's the pathology that's going on here is that there's an imbalance of iron, meaning there's not too much iron. The iron is abnormally distributed within the cells within the nervous system, and that causes a variety of problems. It causes proteins to aggregate. It generates free radicals that damage intracellular structures. So what we're really trying to do is address that iron imbalance. So the group of diseases that we're targeting are referred to as Parkinsonian disorders, and they get their name from Parkinson's disease, which is the most common cause of this motor syndrome that we've probably all seen in friends or family members, or sometimes even older individuals walking down the street. And that's the slow movement, the short steps, the shuffling gait, if you will, or the tremor. And so in addition to Parkinson's disease, which is, affects many people, there are these rare forms called one is multiple system atrophy. What they have is they've got symptoms in addition to the motor symptoms that cause as much or more pathology and more impairment of quality of life. So for example, I won't go through this in detail, but they have difficulty maintaining their blood pressure when they go from a sitting to a standing position. All of us might get lightheaded when you stand up quickly, or when you go from a squatting position and stand up. Normally, in most of us, our nervous systems counteract that change, increase your blood pressure, and you're fine. With patients with MSA, their blood pressure doesn't increase often, and they remain lightheaded. They can't walk significantly. Sometimes they pass out. So it's a major source of disability in these individuals. So they have other symptoms that cause similar amounts of difficulty that I won't go into. So if you actually go to the next bullet, Dan, you'll see that MSA is indeed our lead indication. That's actually the next part of the slide, Dan. And it is rapidly progressive and highly debilitating. I'll tell you that the disease progresses. People probably know about ALS, and I think MSA is sometimes referred to as the ALS of movement disorders. There's a different kind of neurodegenerative disease. And these patients progress rapidly. I'm very sorry to say. Typically, patients go from first symptoms to death within an average of 6 to 8 years. And more than half of patients require a wheelchair 5 years after symptoms first appear. So there's really an urgent need to develop something new for these individuals. So on the bottom part of the slide, the next, if you advance, this basically describes our treatment approach with our molecule 434. We aim to redistribute this excess iron, which is, maldistributed within the nervous system. In doing so, we want to preserve important proteins that are essential for nerves to communicate with one another. We also want to reduce the production of those free radicals. And then the net effect on that is to preserve neurons and their support cells. And in doing so, by preserving those neurons and their communication with one another, the aim is to stabilize or improve function. And that's the overarching goal that we took going into our clinical program. So I think if you go to the next slide, I want to tell you about the trial results that we disclosed in January, and then give you a very brief update. So we enrolled a total of 77 patients. With the criteria that we used to enter them are in the gray box. But basically, they had a clinical diagnosis of MSA, and we did require abnormal biomarker evidence to enroll them in the trial. And this is, I think, a large part of our success because these biomarkers that we required the patients to have are not present in patients with Parkinson's disease. And it's important to emphasize that early on, an MSA patient can look an awful lot like a Parkinson's patient. So we did this to try and make our patient population as homogeneous as possible. And I think that's a large part of why we were so successful. So after these 77 patients were identified, they were randomly assigned to 3 treatment groups: 75 milligrams twice a day, 50 milligrams, or a placebo. And then the key endpoints are listed on the right. The key clinical endpoint is the modified MSA rating scale, and that's a mouthful, I know, so we call it the Unified MSA Rating Scale, or UMSARS. And this is an activities of daily living scale. And what I'll tell you about this is that it assesses 12 domains that are affected in individuals with MSA. So it assesses their speech, their swallowing, their fine motor skills in terms of either handwriting or dressing themselves or even bathing themselves. But it also looks at walking, falling, GI symptoms, urinary symptoms, and those blood pressure symptoms when they go from sitting to standing. Those are referred to as orthostatic hypotension symptoms. So it looks at all these symptoms, and it's a clinician administers this scale at baseline and then periodically over the course of the study. What's important to say about this endpoint is not only does it assess the domains and patients' abilities to function within those 12 domains, it's the endpoint that the FDA and the European Medicines Agency and almost all regulatory authorities are interested to support an approval. And that's why the results from this study were so powerful, because this is the endpoint where we demonstrated such nice efficacy. Okay, next slide, Dan. Before I show you the main result, these are the baseline characteristics. So whenever you disclose a trial, you want to evaluate, are they typical for the patient population? And I'd say they are. If you look at the age, the average age is in the low 60s, and that's pretty typical for an MSA population. They tend to present about 10 years earlier on average than a Parkinson's disease patient. And then the other characteristics are generally well balanced. You sometimes see higher scores in placebo on this UMSARS score or on this motor score. But all the analyses we do that we had planned to do accounts for differences in that baseline UMSARS score. Now, if you look at the bottom of the slide, you see in the pink circle, there are two baseline parameters where there is a pretty significant imbalance between the dose groups. So clinical phenotype means there's two types of MSA. There's the Parkinsonian variant, where patients have the Parkinsonism symptoms we already talked about. But there's another type that's called the cerebellar variant, and that is a different set of motor problems that causes unstable gait, frequent falls, uncoordinated movements, etc. Now, we see that in the highest dose group, the 75 milligram group, that's about 10 to 11% higher than the other dose groups. Similarly, but perhaps a bit more dramatically, if you look at the rate of severe orthostatic hypotension, that's when people go from a sitting to a standing position. If their blood pressure falls by more than 30 millimeters of mercury, which is a lot, that's how we define it. And you see the 75 milligram dose group had about 25% higher rate of that finding at baseline. The reason these two differences are important is that they're both individually rapid predictors of rapid disease progression. And we think this is in large part why the 75 milligram dose group underperforms compared to what we were expecting and compared to the 50 milligram dose group. So if you go to the next slide, which has a few animations, Dan, but I think if we just pause here for a moment, there's a lot going on, so I'll just walk you through it. X-axis is time, and if you look at the Y-axis, that's the change in that unified MSA rating scale. Higher scores are worse. And what you do is you see placebo in the black dashed line. From baseline to end treatment, that increases by about 8 points, which is typical and consistent with other published studies. Now, if you look at the two active dose groups, the pink and blue, you see that they also increase, but they increase much more slowly than the placebo does. And if you note at 26 weeks, 75 milligrams is better than 50 milligrams, which is better than placebo. And what we think is, and then at the end of treatment, 50 milligrams actually looks better than 75 milligrams. And we appear to lose efficacy, a little bit of efficacy at that 75 milligram dose group. If you think back on the other table that I just showed you with the 75 milligram dose group having those two high rates of orthostatic hypotension and that clinical phenotype, that MSA, the Parkinsonian phenotype, we think that's a large reason why these patients tend to appear to lose efficacy and progress more rapidly over the second half year of the study. Now, if you animate to the next part of the slide, Dan, what we see, and just to put these numbers in context, the 50 milligram dose group declined by 3.8 points less than placebo, or 48%, and the 75 milligram dose group declined by about 30% less. both these levels of improvement over placebo are clinically significant. It's been just established and published that a treatment benefit or a treatment difference between active and placebo of 1.5 points is clinically meaningful. So we far exceed that. Now, one thing, and I haven't shown the data, and we're probably going to present it later this year at a scientific congress, or present it at a subsequent webinar, but when you do account for those baseline differences in orthostatic hypotension and the clinical phenotype, this efficacy signal at 75 milligrams increases substantially, and it starts approaching what we see at 50 milligrams. So we think this explains in large part why the 75 milligram dose group appears to be underperforming. As I've said in prior presentations, I am convinced this is a population issue. This is not a drug issue. We see no untoward effects of the 75 milligram dose group in terms of adverse events or lab abnormalities or anything that would indicate that the drug is causing the individuals any harm. I just think they are a rapidly progressive patient population. Okay, so let's go on to the next slide. So that's one important efficacy endpoint, the one the FDA cares about. Now, this is the orthostatic hypotension symptom assessment, and this is what's called a patient-reported outcome. And here's where patients are given a piece of paper, and they're saying at baseline and then every 3 months after, rate your blood pressure symptoms when you go from a sitting to a standing position. How do you feel? And they look at dizziness, vision problems, weakness, etc. And then we look at the end of therapy, and then we compute the change from baseline to the end of therapy. And you see that higher scores here are worse, and the placebo patients decline on average by about 6 points, whereas the active dose groups actually improve a little bit. And you don't expect any improvement in this disease. It really does progress so rapidly. So I think these benefits are clinically meaningful, and I think that's reflected in other efficacy endpoints that we've presented elsewhere. When you look at the clinical global impression from the clinician standpoint, the active dose groups do better than the placebo patients. So if we go on to the next slide, this is a very interesting aspect of the study. We decided, based on what we learned in our natural history study, to look at activity of patients in the outpatient setting, which is very different from what you learn when you examine someone in the clinic, where you're looking for maybe a subtle neurologic deficit. So what people do, and if you can see on the right side of the slide, there's this pendant that's surrounded in orange, and they wear it around their neck for 2-week periods at baseline, and then at 3, 6, 9, and 12 months. And then at the end of therapy, we look at the change, again, like the other scales, we look at the change. So here, if you look at step count and then maybe total walking time as an example, we see that the placebo patients in gray lose about 28 minutes of walking time per day over the course of the year. That's quite profound if you think about it. And then when you look at what's happening in the 2 active dose groups, it's about half that. I don't think you should really worry too much about these p-values. They're starting to approach significance, but in a small study like ours, where it's not really powered, I think it's the magnitude of the benefit that is far more important than whether or not there's a positive p-value. So we see this consistently across these endpoints. And I think this is really important because if you think about the disease, this is a disease where if I showed you, people go from walking normally to walking with assistance like a cane, and then a walker, and then a wheelchair. So if you can slow down their disease progression and you can increase the time that they're walking, you can have a real profound impact on patients' quality of life. Okay, so the next slide. Adverse events. Whenever, I think most drugs, if you look at that are treating a neurologic disease, you do see a lot of CNS adverse effects because they stimulate a receptor, they mimic like dopamine or something like that. We don't do any of that. Our drug only works by preserving neurons, and for that reason, we were really happy to see that we don't see any significant CNS adverse events, and not only that, but we don't see any signal of increased adverse events among active treated patients compared to placebo. And similarly, that's true for individual adverse events, the most common ones listed there, as well as severe adverse events or serious adverse events. And none of those severe or serious adverse events were attributed to study drug. Finally, because the drug does target iron, we are very careful in monitoring the potential for anemia or iron depletion, and we didn't see any of that in the study. So consistent with the moderate affinity, the fact that we refer to this as an iron chaperone is the fact that we don't affect these iron parameters. So very important to have a good safety profile as we've observed. The next slide, I think, talks about some of the biomarkers that we looked at. So because this is a phase 2 trial, we looked at a lot of exploratory endpoints. And we actually selected these biomarker endpoints as key endpoints in the trial. And what we see on this graph, and I know there's quite a bit going on, is we look at the change in iron content, because we do target iron on MRI, at both the 50 milligram dose level and the 75 milligram dose level, both at 26 and at 52 weeks. And what you see is clear evidence of target engagement by reduced iron accumulation in this one region called the GP or globus pallidus. And then at 50 milligrams, we see the same effect in another brain region, the putamen and the substantia nigra. And all these regions are ones that are known to be affected in the disease MSA. So we do think that we see evidence of target engagement, which means that we are really binding what we think we wanted to bind in order for the drug to achieve its clinical benefit. And we did see reduced iron content in this brain region of the substantia nigra at 50 milligrams, but not at 75. We think there are technical reasons why we did not observe those changes at the 75 milligram dose level. Now, the next part of the slide on the right, I think, is some tantalizing data about changes in brain volume. And if you remember, this disease is called MSA, meaning it is multiple system atrophy, which means these regions that I talked about on the left side of the slide, where we looked at iron, those are brain regions that atrophy over time because there is ongoing neurodegeneration, you get cell loss, and the brain actually shrinks. So that's what we're looking at on the right side of the slide. And our colleagues at Vanderbilt developed something called the MSA Atrophy Index, which is really a composite score that looks at the amount of atrophy in the putamen and the globus that are mentioned on the left, as well as two other regions that are affected in MSA. And then we compare the change in brain volume in those regions in our study patients to a cohort of healthy normal people that is publicly available. And what you see, again, here, lower numbers are worse because this is actually reflecting the atrophy. And you see that at 26 weeks, the gray bar, you get atrophy of about minus 0.25 on this Z-score, a statistical measure. Whereas at the pink and blue, you definitely see less atrophy at that time point. At 52 weeks, that trend continues. So I think this is quite important. MRI, even though we are using very sophisticated measurement tools, is using a rather blunt instrument to look at neurodegeneration. And I think this is quite an important finding. So if you animate to the last part of the slide, Dan, you'll see that we feel like we are really showing target engagement by changing iron content in those brain regions, and that we are preserving brain volume, or at least showing trends in preserving brain volume. Okay, so we can go on. I think that is probably what I wanted to talk about regarding the phase 2 trial. I'm happy to talk about anything else. I think I've told you that we are really trying to understand the differences in the doses, and we've largely done that. I think a couple other things just to mention is that we've presented the data 3 times publicly at scientific congresses, one at the American Academy of Neurology meeting in April, and then at an MSA Congress, both in London in late April and then in Boston in early May. The meeting in Boston was a rather large meeting. It was very well attended by scientists, clinicians, and patients. And I've worked in rare diseases before, and I would say that the patient presence there was really important. It's very grounding, and it reminds us how devastated this patient group is and how desperately they need a new therapy. Patient advocate who had received an award for his advocacy over the past 12 months happened to be a participant in one of our trials. He identified himself afterwards. And when he was making remarks in accepting his award, he actually said, in addition to his advocacy work, and he said, I think I would be remiss if I didn't encourage everything we could do to get ATH-434 in everybody's hands, which was really a strong endorsement. And I think it really, that as well as other comments made by other patients reminds us why we're doing this. I think they're so appreciative of how we're working to try and bring a new therapy to these patients. And they're really, they are quite devastated when they get this diagnosis. So I think myself, as well as our entire team, are very gratified, but with the progress that we've made, and we're really looking forward to getting this to the next phase of development as fast as possible. So with that, I'll kind of turn it back to you, Dan. I'm happy to answer any questions you or the audience might have.
Thanks, Dr. Stamler. A great presentation as always, helping us understand very complicated topics into a language we can all understand. Look, we've got a lot of great questions coming in via the chat function and also email. But just to inject my own agenda over the top of this for a moment, I would like to ask you your opinion of the recent study we saw come out of University of Auckland and the University of Sydney, which seemingly could upend the understanding of MSA and the treatment. I was wondering if you could comment on that and just put it into context of your work in Alterity.
Yeah, that was a very interesting publication. For those who may not have seen it, Glenda Halliday, who is a prominent researcher, basic scientist in Australia, as well as collaborators in New Zealand, were investigating the role of aggregated alpha-synuclein in the pathology of MSA. So we didn't talk too much about this protein, but as mentioned, this is a protein that is present in all neurons and really is necessary to allow neurons to communicate with one another. Historically, this disease, MSA, has been thought of as a disease of the support cells, the glial support cells, because what they do is they scavenge that when that protein misfolds and aggregates, the glia cells scavenge it from the neurons to hope to preserve the neuron's function. And people think, and then the glia kind of get overloaded with these deposits of this aggregated protein. So people say that's the disease and that's likely what's causing all the problems. What these investigators showed is that the aggregated alpha-synuclein actually is toxic to the neurons. It actually aggregates and then it penetrates the nucleus and actually leads to neuronal death. And that is actually quite important when you're developing new therapies for this disease, because you want drugs that work inside the cell. And our drug does penetrate the cell. We've shown that in vitro systems. We've shown that in various animal models, that we do penetrate the cell, which means that we are targeting this disease where the pathology is. There are many therapies in development, two prominent ones from two rather large companies, one Japanese and one European, that are antibodies. And antibodies have been used in treating neurologic diseases and Alzheimer's disease. They haven't done so well. I mean, they have a modest benefit. And the same thing, there have been antibodies that are targeted towards abnormal proteins in Parkinson's disease. Those also haven't done so well. Biogen had one and it failed. Prothena had one that had a negative phase 2 and is going on. But I think the point to be made is those antibodies, they have a difficult time getting in the central nervous system, but more importantly, they don't penetrate inside cells. So we think it's quite important that our drug is actually acting on that aggregated alpha-synuclein and really trying to prevent the pathology that's in the cell. And we think that's in large part why we saw such robust efficacy in our trial.
Thank you, Dr. Stamler. Look, I do have a lot of questions coming through, so I'm going to kind of aggregate them and sort of break them down to a couple of areas. Probably the number one question we've got coming in, unsurprisingly, is about timeline. So when is the full data coming out? And when will the post-phase 2, pre-phase 3 meeting with the FDA take place?
All right, the last one, that's a longer answer. We expect that the data will be finalized in next month. That's our goal. We've been a bit limited by the fact that one of the biomarkers that we've included in our analysis, we've had some bioanalytical problems with that. So we are actually re-assaying all those specimens at a brand new laboratory, and that's something that we're putting into place right now. So as soon as we get those data, we'll incorporate that in and we'll run the final analysis. What I can tell you, though, with a high degree of confidence is that we have looked at virtually all of the endpoints, and I continue, because I am I'd say a scrupulous drug developer, I continue to pressure test the data at every turn to make sure that that I'm absolutely convinced that the data is as strong as it appears on first blush. And I haven't seen anything in the data that's changed my mind, either from a safety or an efficacy standpoint. So, but anyway, coming back to your question, I would say in the July timeframe is when we expect the final data. Which is interesting. I mean, I know you didn't ask about this. We're getting final data from our open label study in the same time frame. So we may have two separate disclosures if we have new data, depending on when we get those. So those data are both in process. All right, now the second question, sorry. When are we going to the FDA? So we have several regulatory interactions expected in the next 6 months in different disciplines. So, to get to phase 3 and ultimately to commercialization, you've got chemistry issues, you have preclinical issues, which is toxicology and, say, animal studies looking at blood levels of the drug and metabolites, and then you've got clinical issues. The clinical issues are, I think, you want to address at the end of the process, and that's because you need all this other work in place in order to have that definitive discussion. You want to go to that so-called end of phase 2 meeting where you vet the final protocol with everything else buttoned up. So I'll come back to when we think the timing of that's going to be. But before that, what we have to do is we're focusing probably most of our energy, at least I am, and as a company, on the chemistry and the manufacturing as well as the non-clinical. And of course, we're finalizing the analysis of the data. The chemistry and the manufacturing is going quite well. We've identified a vendor who is making the active pharmaceutical ingredient, or API, and that is in process right now. There's a remarkable amount of detail and complexity associated with that, but we've assembled a really strong team that is managing that for us. So that is going on, but what we'll need to do is we're going to need to go to talk to the FDA towards the, maybe the September, October timeframe, make sure that they agree with our plans, kind of how we're going to actually make the drug and what are our specifications for releasing it and such. So that's one work stream, if you will. Then the next work stream is we're looking at the non-clinical development work. When you do phase 2, you don't have to have every piece of toxicology data done, but we are now doing some additional clinical or additional non-clinical studies that are required to do phase 3, whether it's embryo fetal ones to look at birth control requirements in the course of the study, or whether or not you have to study metabolites in toxicology studies in further depth. So that is all going on right now, and we expect that we're going to be having a regulatory interaction or two with the FDA in the next 6 months on that program. And there's a unique mechanism that we may use to have small targeted meetings so that we can kind of move things along as efficiently as possible. That's really our mission. And then finally, once we get those two sets of meetings, the chemistry and the non-clinical meetings under our belt, that's when we will go and submit the final protocol and have the meeting with the FDA. Now, in the interim, we're doing all the analysis we need to do to understand that 75 if there are any other 75 milligram dose issues, and we're designing the definitive phase 3 trial. Keep in mind, we made some very educated guesses in terms of the design of the phase 2 study. Now we've got a wealth of data. We can understand who progressed rapidly, who didn't, how we want to fine tune our selection criteria. So that work is going to be ongoing in parallel, where we are really looking at every scrap of data and making sure we know everything we can about these patients to de-risk the phase 3 trial even more. That'll be ongoing. And then I would imagine by the end of the year is our goal, or perhaps early next year, depending on how workload is at the FDA and when these other meetings happen, we'll go and we'll have that discussion with the FDA, the so-called end of phase 2 meeting. It's coming out of there that we expect we'll get the green light to go to the next study, and then we'll be off to the races. But the timeline is going to be limited by when we have drug. And that's why we're pushing so hard on everything related to the chemistry and manufacturing, because that has a long lead time. You have a lot of steps that have to happen. So that's why it's getting all of our attention now. But we have our line of sight on that end of phase 2 meeting, which is, if you will, the end of the race. So if you will, I'd say for the next 6 to 8 months, it's more of a marathon than a sprint. I will say to people who are long-term investors, well, besides thank you for your patience, but because we didn't have as much capital as we wanted towards the end of last year before we unblinded the data, we couldn't do all these studies that we're doing now at risk. We did some to try and shorten the timeline, but we couldn't get there as fast as we could if we could have spent more money at risk.
The second most popular question we've got coming in, just combining them all together, I think obviously is, your view on the potential for early sales concurrent with the phase 3 confirmatory trial, accelerated approval.
Yeah, so the accelerated approval is a topic that I look forward to discussing with the FDA at the end of phase 2 meeting. So that's when we'll have a sense. The FDA, I've learned, as enthusiastic as I am about the data, is hard to predict. But I think if ever there was a good case to be made, I think we've got it. We've shown that 434 works in these 2 dose levels. We've shown the safety profile is benign. We've shown that we've got ancillary endpoints, whether it's the clinical globals, the patient-reported outcome on orthostatic hypotension symptoms, the wearable sensor showing increased activity. We think that's all very powerful. I think what it comes down to, and it's hard to get inside the FDA's head, is how do they feel? How depending on how well we explain what's happening at 75 milligrams, that's going to be a large component as to how they respond to the application. So I think it's a distinct possibility, but it's very hard to handicap. If anyone is a golfer out there, it's hard to know what the FDA is going to say. But we are highly motivated to do that, for not just for shareholders and investors, but also for the patient population. There really is an urgent need.
And bearing in mind, we can't predict the FDA, as you say. Left to you, what would a phase 3 look like? What do you think a phase 3 should look like?
Yeah. So it would be a two-arm study. I would pick, and everyone, it's people say, oh, would you pick the 50 milligram dose? Well, no, because as I've said before, I think it's a population issue, not a dose issue. So I would use the 75 milligram dose group. Obviously, the FDA may have something to say about that. But I would do a 2-arm study, a minimum of 80 patients an arm. And depending on if you actually take the treatment effects that you've got up there on the slide, Dan, actually, if you power a study for the average of those 2 treatment effects of the 3.8 versus 2.4, that's around 3 to 3.1. That's a sample size of about 80 per arm. If you want to be a little more conservative and power just for a treatment effect of the 75 milligram dose group, that might be more like 100 an arm. So I'd say anywhere between 160 to 200 patients total. would be my expectation for the study. And the primary endpoint would be the modified UMSARS as we evaluated in this study.
And we haven't got long to go, so I think the next sort of most popular question we've got coming in, and apologies if I don't get to all these questions, but I guess putting my investment banker hat on, I mean, big pharma. There's a lot of excitement clinically about this. So it's logical that big pharma will be looking. I mean, what's the prospect for JVs, partnership? Perhaps you can't talk about it in detail, but what do you think is the right path here for Alterity?
Yeah, it's an excellent question. There's been strong interest in these data since we released it. There are a handful of companies that we've been talking to, some for years, some for just a year, some for just a few months before we release the top line data. But I can tell you, without actively pursuing any partnership discussions, coming out of these data, we had solid organic interest, meaning they came to us, of 6 companies, of everything from very big to medium sized to small. So there is a high level of interest. Those discussions have been ongoing, and they continue to remain interested. They're interested in the final data, and we meet with them a lot. They ask us a lot of questions, and so I think there's a high level of interest. Now, I just came back last week from the Bio International Partnering Meeting, which is the annual pharma partnering meeting that there are thousands of companies there. So we met with some of our longstanding connections and updated them. But we probably made 6 to 8 strong additional meetings just based on most of those are inbound requests from other companies that are aware. And there are many companies, both big and small, that are really eager for an opportunity like this, especially in a rare disease is very interesting. And neurologic disease, there's great interest. So I'm quite confident that we can find a partnership that will be ideal for shareholders. We can bring in the capital that we need to complete phase 3 without, ideally without raising further capital. But we don't know. We don't, obviously time will tell. Most importantly, we have to have, if we're going to partner, we have to have the right partner, someone that makes sure that we have a seat at the table, that we're developing the drug with them. We've learned a tremendous amount and know how to do these studies. So we want to partner with someone that not only has the resources to commercialize this drug, but is also a good development partner, because many drugs, if you hand them over from small pharma to big pharma, they can, those transitions can sometimes not go well. We want to make sure that doesn't happen for patients and for shareholders. So that's the next step. I spend quite a bit of my time talking to these partners and nurturing those relationships. And I think I'm quite optimistic that we can find the right partnership, and that would be excellent for shareholders and excellent for patients.
Look, we are over time, and I might squeeze a couple more questions in if you can spare the time, Dr. Stamler.
Sure.
Look, I guess speaking of commerciality, commercial assessment, obviously all of us are now sort of looking back through historic pricing of orphan drugs and those sorts of things, and the incidence of the disease, trying to sort of get a handle on this. What work is going on inside of Alterity in terms of the commercial understanding of the potential therapy?
Yeah, so we've engaged an independent firm to do a commercial assessment for us that does this all the time, that has commercial roots as well as strong connections to marketing research. So that study we embarked on in the February-March timeframe. It's 2 parts. There's a qualitative part where they did one-on-one interviews, and then kind of roundtables with a few physicians of differing disciplines, both general neurologists, movement disorder neurologists, autonomic nervous specialists, to understand what they thought of the data, what they thought of the clinical need. We then took all those interviews, and the feedback was very positive, as you can imagine. We took all that and then created a discussion guide where we actually did then moved from a qualitative to a quantitative survey that is wrapping up, that was in the field and will be wrapping up soon. And that's where we'll interview, again, a blend of mostly movement disorder and general neurologists and some autonomic nervous specialists. And the data that we generated will be tested, as well as other relevant data that are available for phase 2 assets that are in development. So I think sometime in I say the summer, our summer, but in the probably in the July August timeframe, we'll have some data to share with the market, and we welcome we welcome sharing that transparently because we think it's quite important. I'll just mention that our peak sales estimate that is currently part of our deck was done a few years ago and was updated back in 2023 for updated prevalence and pricing information. But that was still based on a target product profile that was rather modest. We made some assumptions about improvement in orthostatic symptoms, no assumption about improvement in the UMSAR scale. And we still got estimated peak sales of over $1 billion. So just in the US. So I think I'm expecting a robust increase, and I'm really looking forward to getting these results and sharing them with you.
Look, just two more questions, so I'm going to stretch just a little bit. Look, I guess the next question is a little potentially unfair, because I guess you can't answer it. But I'll just ask you to speculate about the success that we've seen with the therapy to date for MSA, and how can we? What's a sensible extrapolation that we can think about across other neurological conditions in the Parkinsonian family? Is there anything that you can say that would give us any idea of how this therapy could be used more broadly?
Yeah, I mean, I didn't kind of dwell on it, but we know that MSA, and let's take Parkinson's disease as an example, but there are other diseases that that are similar. They have similar underlying pathology, meaning they have elevated iron in areas of pathology. They have misfolding alpha-synuclein and aggregating alpha-synuclein. So there is good reason to believe that 434 has potential to address those diseases. But they're not the same, right? The pathology, they progress more slowly. So that's why you need to do bigger trials, longer trials, as opposed to a 12-month trial. You might need to do 18 months or even 24 months to demonstrate benefit. But I would just remind everyone that we've historically done a lot of preclinical work in Parkinson's disease, several animal models, and I think this may be in our corporate slide deck that's on our website. Various, like 3 or 4 different models, both with different genetic defects, different toxin models, and most recently a primate study that looked at functional performance that is much closer to humans and than the rodents. And we've seen kind of really nice efficacy across the board. So I think there's good reason to believe that we have potential in Parkinson's disease, and I think it's something that we're going to evaluate doing clinical trials. I certainly know, and I don't know if this is going to come out of the marketing research, whether or not what clinicians feel about its applicability. I think people who are knowledgeable about these diseases know that alpha-synuclein and iron are both factors in Parkinson's disease as well as MSA. So I'm encouraged, and I would really look forward to investigating this further and doing a proper trial in Parkinson's disease.
Look, we might have to leave it there, but we've run well and truly over time. Just a couple of adverts though. Alterity, Dr. Stamler and the Alterity team will be on the road for a non-deal roadshow during August. So I'm sure the relevant people will be in touch. And Dr. Stamler, you'll also be presenting at BioShares. Is that correct?
That's correct. That's correct. We're going to participate there.
And I believe Dr. Daniel Claassen will be
That's right.
attending.
Yeah, Dr. Claassen is our principal investigator. He was the one who oversaw the neuroimaging as well as the clinical trial implementation. And he is expected to join us on that non-deal roadshow in early August. So
Yeah,
that'll
and
be fun. That'll be great. That'll be great to get his clinical perspective.
fantastic. Yeah, and look, our channel checks have found that Dr. Claassen's a bit of a rock star in the neurological arena. So it'd be great to have him on board and hear from him. Dr. Stamler, thank you for your time. I appreciate you allowing us to go a little bit over time. Thank you everyone for joining. And as I say, everyone will be in touch for the various investor events that are coming up over the next few months. Thank you again.
Great. Thanks for joining. Bye-bye.